DNA barcoding and its applications in genetic, species, molecular diversity, and how it is related with taxonomy. Let us see with that. Most of you people are well aware about barcoding. Generally, barcoding is done to identify the things in general. Similarly, in biological systems, DNA sequences are used as DNA barcodes. Let us see what is a DNA barcode. You know, all genes, all genetic material is made up of DNA. The DNA has double-stranded nature, it's helical, and each DNA strand is composed of several nucleotide sequences. So out of these, so many nucleotides, there are certain genes common to all species, certain genes. Either it may be plants, animals, or anywhere. In almost all species, some common genes are there. For example, plants are green in color. All green plants have plastids, chloroplasts. And you know that chloroplast itself has a genome, which we call as a chloroplast genome or CP genome. So here DNA barcodes are standard short sequences of DNA which are between 400 to 800 base pair long, that in theory can be easily isolated and characterized for all species of plants on this planet. So DNA barcoding are certain specific species of sequences of nucleotides can be used for identification of species using a short section of DNA from a specific gene or genes. For example, here below I have shown a small sequence here. This you can say this is nucleotide sequence. You know, all nucleotide sequence are made up of AGCT. A stands for adenine, G for guanine, C for cytosine, and T for thymine. So all these DNA sequences, they are made up of the DNA. These are something like 460 sequences are there. All these nucleotide sequences are presented here in FASTA file format. Now, for example, if you take any species, any plant, as I told you, the chloroplast contains a DNA, chloroplast DNA. In the chloroplast DNA, there are certain barcode genes are present. What are these barcode genes? Barcode genes, they contain at least two short regions that are highly conserved between all species. For example, I am talking about a gene which is known as X gene. That X gene must have a conserved sequence. Conserved means after so many thousands of years also, that same sequence is conserved, retained as it is, without much mutation or recombination, etc. Some sequences of DNA are preserved thousands of years, and these are called as conserved DNA sequences. Here, below I have mentioned, there are some commonly used barcode genes are there in plants. One is Rbcl gene. Rbcl, it is a chloroplast gene. It encodes for ribulose, bisphosphate, carboxylase, large subunit gene. So this gene has two terminal ends. Both the terminal ends have got highly conserved sequences. In between these two conserved sequences, the DNA sequences, they most frequently they go on changing, mutations occur, recombination occurs, something like that. Due to that, the intermediary sequences they shows variation. So here barcode gene, the what we call imaginary barcode gene, has on both the sides short regions that are highly conserved, but between these two highly conserved regions, there is long, some 600, 800 nucleotides are there. They are free to undergo mutation. Mutations occur. That is why, just like our fingerprints, all plant Rbcl genes, they do not match with each other. At least in the intermediate gene, one or two changes will occur in nucleotide sequence. So the nucleotide sequence, they vary with each other. They show variations. But how many nucleotides are changing? Only one or two are changing. 98, 99% DNA sequences are same. It means both the species, they are sister species. They have evolved from a common ancestor and they show very closest relation. If the DNA sequences are more varying by 50%, 60% like that, these two species, they do not have close relation. They are different, distant species. They may not be related with each other. So in DNA barcoding, we use certain barcoding genes. 3 barcoding genes are most commonly used in plants. First one is Rbcl gene. Just now I talked. Second one, COX1 gene, C for cytochrome, OX for oxidase. First region of that gene, COX1 gene, it is present in mitochondrial cytochrome oxidase 1 gene, and it is the most commonly used marker or barcode gene. Suppose if you want to go undertake barcoding studies in animals, there is no use using Rbcl gene. The animals doesn't have any plastids, no chloroplast, no cpDNA. So there you cannot use Rbcl gene. Then which gene you will be using? COX1 gene, mitochondrial cytochrome oxidase gene, is the commonly used barcode gene in animals. Even that can be used for plants also. In plants also mitochondria are there. So it is a common barcode gene for both plants and animals. Similarly, the third one is MATK gene. It is also another chloroplast gene present in cpDNA. It is exclusively used in plants only. MATK gene, the full form is maturase K region of the gene found in chloroplast of most of the plants. So these three are the most common barcode genes. So if you want to take any barcode gene, it should have certain specific characters. A barcode gene. What are those specific characters? It should have on both the sides, extreme left side, extreme right side, it should have two short regions which are highly conserved in all species. In most of the species, it is conserved. Second one, the intermediary region that is prone to mutation. It is free to undergo mutations. So it is variable region goes on changing. The third one, if the conserved region is there, you can, by doing in situ hybridization, by sending some markers, you can catch hold of these conserved regions. You can identify that Rbcl gene or COX gene, like that. For that, they have separate markers are there. We'll be seeing around this. Let us see, in plants, the most commonly used is Rbcl gene. As I told you, Rbcl stands for ribulose 1,5 bisphosphate, biphosphate, carboxylase. This is an enzyme. This enzyme is a protein. This protein is made up of two subunits, one small subunit and one large subunit. So here for in Rbcl gene, L stands for large subunit gene. So this is the most widely used barcode gene in plants. Here I have mentioned some points. Chloroplast genome, generally we called as the ctDNA. CT stands for chloroplast, or cpDNA, that is chloroplast DNA, chloroplast genome. In plants is a circular double-stranded DNA molecule. It is present in the cytoplasm, especially in the chloroplast. It has the genome size, if you see, up to 2000 kb, kilo nitrogen bases, kilo bases we call. So that much reasonably big genome is there. Genome means DNA molecule. The chloroplast have a circular DNA molecule, which is called as its own genome, chloroplast genome, which is 85 to 2000 kilobases. This chloroplast genome, it codes for many enzymes. We know one gene, one enzyme concept. If it is coding so many enzymes, that means it has so many genes. So one of those genes is ribulose 1,5 bisphosphate carboxylase oxygenase. In short, we call Rubisco. Ribulose, RU stands for ribulose, bis stands for biphosphate, bisphosphate, and C for carboxylase and O for oxygenase, which plays an important role in carbon fixation in photosynthesis. Without this enzyme, probably no chloroplast can fix carbon dioxide. Even though chlorophyll is there, carbon dioxide is there, sunlight is there, water is there, everything is there. If this enzyme is not there, that carbon dioxide cannot combine with the other molecules, oxygen and like that, water. So this enzyme is made up of two protein subunits, as already I told you. They are called as LSU, large chain subunit, and small subunit, small chain subunit. The gene responsible for synthesizing the large subunit is called as Rbcl gene. I hope you got me, you understand what I'm telling. Here, the large subunit has coded by something like 1400 base pair long Rubisco Rbcl gene, the large subunit. Rbcl region is highly conserved and universally present in almost all plant species. So these two criteria, just now I told you, if you want, you cannot use any DNA as a barcoding gene. Barcoding gene should have certain characters of its own. So the first and foremost important character is it should have conserved regions. This Rbcl region has highly conserved regions on both the ends, and these conserved regions are universally present in almost all plant species without any exceptions. Another superiority of Rbcl gene is its low level of mutation. Spontaneous mutations occur in most of the DNA. Rbcl gene shows low level of mutation as compared to other genes which show mutation rate in hundreds and thousands. So compared to that Rbcl gene has low level of mutation. Because of its universality, it is universally, everywhere it is present in all chloroplasts, and ease in amplifying and analysis, it is used as the barcode marker. This chloroplast sequence is widely used in DNA barcoding studies. I think these 2 points should be made together, not two separate points, it's a single point. So due to these reasons, this chloroplast gene is widely used in DNA barcoding studies. So this is also known as barcode DNA. Why is Rbcl used in barcoding? What are its specific characters? Means we have certain reasons for that. Rbcl gene serves as an ideal barcode because it codes for part of the key photosynthesis engine, that is ribulose bisphosphate carboxylase. It is present virtually in almost all plant species. One section of its DNA sequence is very variable between species, making it ideal for DNA barcoding. So it can be used to distinguish plants with the same genus. Within the same genus, if we have 20 species, 30 species like that, all species, they share the same type of bin with few variations in nucleotides. So it becomes an ideal barcode. In 2009, the Consortium for the Barcode of Life Plant Working Group proposed the chloroplast gene Rbcl as the core barcode of plant species. So hence, afterwards, Rbcl is widely used as a barcode gene for plant analysis, phylogenetic analysis. Since Rbcl is characterized by its universality, easy amplification, and compatibility, this gene has been proposed as the barcode fragment. So the DNA nucleotide sequence, it may be some 500 sequences, 600 sequences like that. All these sequences, earlier I have shown you, the sequences we cannot remember. For example, in the last slide I have mentioned, this is the DNA sequence of a particular Rbcl gene of some species. No, you cannot simply remember TT, GA, like that. 500 sequence in the same sequence we cannot remember. So then how to preserve this? They have the computer people, they have discovered a format to remember this. That is a simple word format, and which is called as FASTA format. So the name FASTA derives from a software package written in the mid-1980s that searches quickly through large collection of sequence data. This sequence data may be DNA nucleotide sequences, or if it is protein, proteins are made up of amino acid sequence. For both it can be used. So this software is called as FASTA. It is in their computer language. It is the most basic format for the storage of DNA and protein sequences in text-based format using single letter codes. Single letter A for adenine, C for cytosine, like that, or phenylalanine, PHE, Arginine, ARG, if it is the amino acid summary. So the connotation FAST-P, FAST-N, and FAST-A also you come across the literature. Now, they denote, P denotes proteins, FAST-P means it is for protein sequence, FAST-N is nucleotide sequence, and FAST-A, it denotes all means both nucleotides as well as proteins. So generally people prefer fast A sequence instead of discriminating these two. It is very easy to store in the form of the computer, store in a computer. The fast files can be opened in almost any text editor, even in Word also you can open that. How to make a reference barcode? Suppose if you want to make a barcode, you want to identify a species, you found a new plant. Now you are unable to identify that plant. Most of the plants using Bentham and Hooker classification or something like that, you are confusing whether this plant belongs to this genus or species. Yeah, if the genus is the same, whether it belongs to this species or the other species. If you want to find out, that molecular analysis is the very accurate, most reliable method. So molecular techniques are used nowadays. When conventional methods, morphological methods and other methods, they fail, yeah, they are not sufficient, then people resort to this molecular biology techniques. So here, DNA barcoding, since the word is DNA, we are extracting DNA from the plant, we are no way concerned with proteins. So first you have to extract genomic DNA. Why the word genomic DNA? Genomic DNA is the DNA which is present in the whole cell, total DNA. Either let it be a chloroplast DNA or it may be nuclear DNA, so the total DNA, that is called a genomic DNA from the leaf tissues. Leaf tissues are crushed, and you know how to isolate DNA. In MSc also you have isolated DNA and all. So here I cannot mention that pure almost complete method and all. So extraction of genomic DNA from the leaves is carried out. Once the DNA has come up, it is digested with restriction endonucleases. So to make the long, enormously long DNA into small fragments, these fragments are amplified with the help of PCR. The PCR amplification of the isolated genomic DNA and PCR amplification of the markers. I told you what is the marker? Marker is a particular sequence of DNA which can go and bind with your Rbcl conserved region. So it will make a double-stranded region, some part. When it is filtered through hydroxyleptide columns, all single-stranded DNA will go out, filtered. Only this double-stranded piece that will remain in the hydroxyleptide columns. So identification of Rbcl genes and isolated genomic DNA using this technique, hybridization, in situ hybridization technique, their isolation and purification, that will be the next step. DNA sequence analysis of Rbcl barcode genes present in the genomic DNA and generation of nucleotide sequence in FASTA format is done on automated DNA sequencing machine. So DNA sequencing machine is there. After PCR amplification, the amplified Rbcl genes that is fed into automated DNA sequence machine. That sequence machine produces data in the form of AA, CC, DT, TG, like that. And later it is formatted into a FASTA sequences. Once you have created FASTA sequences, that means you have a reference barcode. Now this you can use to identify your unknown species. There is a molecular software is there. That software is called as Mega Molecular Evolutionary Genetic Analysis. Mega stands for what? Molecular Evolutionary Genetic Analysis. Now the latest is the 11 series. That software you have to open. It is free to students. Once you open the software, most of the things are self-explanatory. And whatever your FASTA sequence is there, you have to load FASTA sequences into that machine, into that software. Automatically, it will take up that, and within few seconds, it will give your this sequence Rbcl barcode which we have created is similar to the already created and stored reference barcodes are there. Immediately it will give a list of 1,000, 2,000, 3,000 plants. This is similar to this is 99.99% similar to another. Third one, it is 98.99% similar, like that. So the highest similarity, 100% similarity means you have found out that species. It will give this sequence is 100% similar to Palana plant, XYZ plant. So you have identified that plant. Or if you cannot identify, let us say it is a new species you have discovered. It has a new DNA sequence which is not already fed into the computer. Already it is not present in the reference library. Then it will give similarity. It is 99.9% similar to this species. That means you have found out its sister, its relative, nearest relative. So that way you can find out the nearer and dearer to that new species. So once we have this analysis in Mega10 or Mega11, so Mega11 can also be used for using its phylogenetic tree, evolutionary tree, from which species it has evolved, what are its ancestors. Suppose I have 20 genus is Desmodium, for example. I have a Desmodium genus. In Desmodium, maybe 13, 14 species I have. I want to find out what is the relation, interrelation between these. Then all 14 FASTA format I will obtain for 14 species. All 14 species I will enter into the Mega11 format, what we call format, and that software I will find constructed tree, phylogenetic tree, based on their relations, evolutionary relationship, how much similar, how much distant they are. Construct a phylogenetic tree. Only a click of the button after feeding what sort of tree, if you want rooted tree, you want not rooted, how you want to, whether cladogram, phylogram, what you want. If you give all this information just by pressing a button, immediately that phylogenetic tree will be constructed. Just now I told you the reference libraries. You have constructed a reference barcode, and these reference barcode you have to compare with the reference libraries. Where are these reference libraries? These reference libraries are available online. Your Mega11 software has all these reference libraries inside it. So these reference libraries are also called as databases. They contain nucleotide sequences or DNA barcodes of previously identified taxa, let it be plants, animals, or any, all, whatever. I don't say it is work is complete, almost all barcodes are present in their database. It is not even 20%, 30% complete. So people, more and more databases are poured every day. If you have a new database, you can submit it to the data banks. Here below I given GenBank is a reference library or a database. EMBL, DDBJ, PDB, all these are data banks. So the sequence of unknown DNA barcode is then compared with those of reference libraries to identify the species. Some well-known reference libraries or databases are GenBank, a genetic database. GenBank is a genetic database that serves as an annotated collection of DNA sequences. So many DNA sequences. Suppose if you are a researcher, you found out a RBL sequence of one species, you can submit it to the gene bank. It is GenBank. Okay. Similarly, European Molecular Biology Laboratory is an intergovernmental organization dedicated to molecular biology research. This also has very huge databases, any type of databases. Suppose if you ask for particular gene, I want a so-and-so gene, I want the nucleotide sequence of a particular gene, immediately you will get it. Most of the genes, their sequences are stored here. DDBJ, DNA Data Bank of Japan. It is a public database of nucleotide sequences established at the National Institution of Genetics, NIG, of Japan. Similarly, PDB. PDB is a protein data bank. It's a database for three-dimensional structural data of large biological molecules such as proteins and as well as DNA also. Sometimes DNA three-dimensional structures is also available in this. So these are the some reference libraries. So what we have learned so far, what is a barcode? What are the specific characters it should have? How to construct a reference barcode? Then what are reference libraries? And how can you compare with this using Mega11? Suppose here a two barcode nucleotide sequences for two species of Desmodium. One is Desmodium elicicarpoides, the first one, and second barcode sequence of Desmodium triquetrum is another one. So these two are sequences of one PhD student. So see here, both the barcodes, when you see, you don't get any idea, all are looking like TTT, GGG, C, etc. But the first one has number of nucleotides is 506. The second number of nucleotides are 527. Why this number? This is Rbcl gene. This is here assembled FASTA format sequence for Rbcl gene. This is also Rbcl gene and this is also Rbcl gene. But the length is not the same, number of nucleotides. They are less here and they are more here. That means some nucleotides are deleted. Some nucleotides are deleted. See what happens if some nucleotides are deleted? It will lose certain characters. If that are very essential character, then Rbcl gene will lose its character. If it is non-essential region, it doesn't matter. So Rbcl will still function with little anomaly. And depending on these two, we will see whether they are related with each other or not. Here both belongs to the Desmodium genus. Both are different species because they have a different Rbcl sequence here, the Rbcl barcodes. So what are the applications of DNA barcoding? Main application of DNA barcoding is identification of species. So you may tell, species I can identify orally and just by seeing its morphology characters and all. But molecular methods like DNA barcoding has great applicability in identification of species when traditional methods are not applicable. Specific short DNA sequences or markers or barcode, they form standard region of a genome which can provide a DNA barcode for identifying species. So DNA barcoding is an invaluable tool to analyze diversity occurring at a genetic, species level or molecular level in living organisms. So the DNA barcoding variation may be present at genetic level, species level. The species may differ. At molecular level, even within the species also we have cultivars and others. At that level also molecular differences may be there, cultivated varieties, etc. So these small differences occur through mutation and recombination mechanisms. Finding out evolutionary relationships between the species belonging to a genera and building a phylogenetic tree to elucidate their evolutionary relationships between closely related species is possible only when you have the barcode regions, nucleotide sequences of certain conserved regions, conserved genes. The gene which is universally present in all plants, such one. So DNA barcode markers can be applied to address basic questions like systematics, ecology, evolutionary biology, pedigree construction, biodiversity, and construction. These applications you will study when you study about all these, how molecular approach can be used in evolutionary biology. Evolutionary biology means in the plants, animals, or anything, how they are progressing, how they are evolving, in which direction, and that explanation is given at molecular level. Identification of endangered and threatened species. Sometimes, if you say some species has extinct, even extinct species also, and if you have a herbarium for that extinct species, people are taking that dried plant leaves from the herbarium. They are isolating DNA, isolating whatever the Rbcl gene is present. They are finding out what is this species, what is it genus. If it's something new, some new name here in herbarium sheet, name will be given. They will find out whether it is a ancestral plant, in which age it is present, a most recent one, recently extinct, like that you can find out. So identifying even extinct plants or endangered or rare and threatened species also can be used. Similarly, in case of animals, linking egg and larval stages. Larva will be there, egg will be there. Often it becomes the eggs are belongs to which, larva belongs to which species, and this adult. So if you want to match these three, you can use barcode, DNA barcode studies. Now, securing intellectual property rights for bioresources. Suppose you have discovered a genetically modified, you have done genetic engineering studies, you have introduced some new genes, and you have find out really a novel plant, and you have the rights, intellectual property rights you got for that plant. If some people are claiming that it is I have discovered and all, it's a different plant. So you can identify these two plants by using the DNA barcoding techniques. It is the same plant or it is a different plant. Forensic science also. In forensic science, suppose a child belongs to this parent or a different father. People that say DNA fingerprinting technique, which also comes under barcoding. So who is the biological father? This father or that father can be like that, can be found out. So in forensic, several applications are there. So these are the few applications of DNA barcoding. Next one, I have taken a screenshot of Mega. I have written Mega11. I think it is Mega10 software. So below this symbol, you can see this symbol. It's like 10, Mega10. So here, if you see this file will be there. Align. Align means whatever the DNA sequence you have isolated after PCR amplification and that DNA is aligned. Aligned means we place side to side this sequence, and the computer has thousands of similar molecules. So it will align, it will compare with each and every molecule just by placing it next to it. And the percent align, how much percent it is similar, 99%, 98%, how much it will tell. After that, you can ask this one through blast analysis. In alignment, the blast analysis will be there. If you do blast means the basic local alignment, something is there. If you do that, it will tell this your DNA molecule, barcode molecule is similar to how many already in reference library you have, it will tell. So where you are, identical molecule you are getting, you are not getting. So that it will tell. After that, directly we will go to this phylogeny. In this phylogeny, it will try to construct it. You have got so many species, not one species. If only one species there, in this align you can find out this species is related with what. If you have several DNA barcodes, just like in I told you in Desmodium, 14 species, one research student has done. If they are to be compared and the evolutionary phylogenetic tree is constructed, then you have to go to phylogeny. All these 14 sequences, if you give, it will compare one with each other and it will prepare a phylogenetic tree. Okay. Similarly, you can find out ancestors who are it's supposed to be imaginary ancestors for that. So like that, in this software, you have several molecules with each other. You can combine with them. So this is the Mega11 software. For students, it is free. You can download even now also. After downloading, you can take certain sequence others. Sequences are also available online. And that sequence, you can find out this sequence is of which species immediately. So just like a practice, I strongly suggest you take certain DNA sequences or given some DNA sequences to here. And these two sequences can be found out with this phylogeny, with this Mega11 software. And you can find out the amount of relation present between these two species. So here, when you have analyzed several species and you are finding out phylogenetic relation, you have to make dendrograms. Phylogenetic trees. The phylogenetic trees, they tell what is the ancestor, what is the second generation, how they are evolved, what is the third level, their evolutionary relationship, fourth one, fifth, and the recent what are the types, like that. For example, if you see here, this may be first evolutionary phase, then the second here, this is called a node. Here one node will be there, This is the ancestor. From this ancestor, two nodes have come, this and this. Means two plants have originated by changing mutations and recombination. From this node, another plant has evolved, and from this, B and C have evolved. So these are called branches. This is another branch, and both the branches are meeting at this node. This node is an ancestor for this B and C. So this node and this one, A, are evolved from this node. This is the common ancestor for A and that imaginary X. So like that. So here, phylogenetic trees are two types. You have a cladogram and phylogram. Both are common name is dendrograms or phylogenetic trees. What is the difference here? You can see all A, B, C, D are in a single line. Single line. Why they are in single line? The branch, it doesn't mean anything. Just to show you have written. You cannot measure it. Its length will not tell you anything. Whereas here, they are not in same level, but branch lengths are changing. It has a very long branch and this has a short branch. This branch will tell how many mutations that Rbcl gene has undergone from this imaginary common ancestor. So after very less mutations, this species has originated. After so many nucleotide mutations present in Rbcl gene, this species has evolved. So if the branches mean something, here are written, the amount of evolutionary divergence, it is. So phylograms are scaled. Scaled means you can measure them, which means that branch lengths are proportional to the amount of evolutionary divergence. In the above cladogram, it does not mean branch length do not reflect the amount of evolutionary divergence between taxa. So this is the small difference between a cladogram and phylogram. So using this, one can prepare either cladogram, phylogram. Only in Mega11 software, you have to instruct, you want a cladogram, a phylogram. So usually people prefer phylograms because it gives evolutionary information. So I hope you understood. Any doubts are there, you can ask me also in between. Anyway, probably this is the last slide. Construction of phylogenetic tree using Mega11. So the 13 or 14 Desmodium species are the barcode region, Rbcl barcode has been fed into the computer. After feeding that, phylogenetic tree, evolutionary tree is mentioned here. Here, can you tell me whether it is a cladogram or phylogram? Here, all branches are not same. So here, the branch is small here, this branch is large, this branch is small, this is another small. Even though letters are mentioned in a single line, the branches are there. Now here we call this 76, 98, these numbers are known as bootstrap values. That also the computer will tell. What are these bootstrap values? If suppose this diagram, if I, let us say this 3, if I repeat the experiment 100 times, 82 times, you will get the same diagram. So reproducibility is more. So in this, generally, the science does not take 100 times, 82 times, 82 times. It will get the same diagram. So reproducibility is more. So in this, generally, the science does not take 100 times, 82 times, 82 times. It will get the same diagram. So in this, generally, Below 50%, they does not take, but one exception you can see 29 years mentioned. Okay, but that limit you can fix. Then phylogenetic tree, this phylogenetic tree is evolutionary. You can see this is the very, very primitive species is Gangeticum. Gangeticum is present for several years. Suppose if you write vertical lines like this is maybe 10,000 years ago. This maybe 9,000 years ago, 8,000 years ago, 7,000 years ago, 6, like that. And this is the most recent one, this line, most recent, this line. So each vertical line indicates how many years back, how many thousands of years back this ancestor was there in existence. This ancestor has converted into these two, and its existence stopped. No more that species is there due to recombination, mutations that has extinct and become two. Okay, so like that. So the lengths of the branches represent the substitution rate, which is defined as the percentage of substitution site for alignment length. So this is how these barcode nucleotide sequence data can be used in finding out evolutionary relations between this. Thank you.